Welcome to iiocs.com Food Tech Club. Today's topic is Principles and Methods of Food Preservation. Let us first see about principles of food preservation. Food spoilage is the process of food becoming unsafe or unacceptable for human consumption. Spoilage is normally caused by the growth of microorganisms. Other losses in quality are caused by natural activities in fresh food tissues. For example, the excessive softening in overripe fruit caused by fruit enzymes. Spoilage and quality losses are partially or completely controlled in properly preserved foods. Food preservation is the maintenance of safe and nutritious food for an extended period of time. Therefore, food is spoiled basically by the microorganisms or any other chemical activities happening inside the food, like ripening or senescence and other metabolic processes happening in the food. Okay, so if we properly preserve the food, then we will be able to delay or stop such activities which cause the food spoilage. Okay, let us see various principles behind preserving the food. So there are three main principles involved in food preservation. First is prevention or delay of microbial decomposition. The next, prevention or delay of self-decomposition of the food, that is basically by enzymes or metabolic processes like respiration and all in the food. Then prevention of damage because of insects, animals or other mechanical causes. Okay, let us see about all these principles one by one. Okay, first is about prevention or delay of microbial decomposition. So under this we have four more categories, so four more methods. So first one being the aseptic method, wherein we keep the food out of microorganisms. Okay, so first method is by keeping the food out of microorganisms. Second is by removing the microorganisms using filtration method. Third one, by hindering the growth of activity of microorganisms. Fourth one is by killing the microorganisms. So let us see the first one. First one is by keeping out the microorganisms, and this process of keeping out the microorganisms is called as asepsis. So let us see what is asepsis. Asepsis refers to keeping out the microorganisms from the food by making use of either the natural covering or providing artificial covering around the food. Okay, so in asepsis what we do, we completely avoid the contamination of microorganisms, food with the microorganisms. So food should not be coming in contact with any kind of microorganisms. So this is called as asepsis. Natural barrier in foods include outer shell of nuts, you can see here, then skin or peel of fruits and vegetables like in oranges, then shell on eggs, or the skin of fat in meat, or the husk of corn, ear corn. Okay, so all these are natural barriers for the microorganisms or any external contaminations. Okay, so similarly we can create an artificial covering for the food, okay, by giving a proper packaging. Similarly, packaging prevents entry of microorganisms in the food. Okay, we saw about asepsis. The next method is filtration method. In filtration, as the name tells, so we filter the food material, especially the liquid food materials, in order to separate the food from the microorganisms. Filtration of liquid foods through bacteria-proof filters is a common method for complete removal of microorganisms from the food. Okay, so it helps in completely removing the microorganisms from the food. Liquid foods are passed through the filters made of suitable material like asbestos pad, diatomaceous earth, unglazed porcelain, etc. are allowed to percolate through either with or without nanofiltration or other filtration method, etc. works on this principle. Okay, so you must have been studied about reverse osmosis or microfiltration or nanofiltration. So all of those filtration methods use such membranes like asbestos pad and all, so in order to filter the microorganisms. Okay, centrifugation, sedimentation, trimming and washing, etc. can also be used but are not very effective. Next method is by hindering the growth and activity of microorganisms. So here also we have different methods, like by using low temperature, by drying the food, by creating anaerobic conditions, or by using chemical preservatives. So first one is by using low temperature. How can we hinder the growth and activity of microorganisms? So microbial growth and enzyme activity is retarded in foods by storing them at low temperature. Okay. The food commodities can be stored under cellar storage. So this is the normal storage method we use in our homes. So that is, we keep certain foods in cool regions, okay, in order to certain semi-perishable or non-perishable foods in cool regions. Okay. Example, onions and potatoes, you must have seen that. You don't keep it in a refrigerator, you keep it outside in a cellar storage. Next one is refrigeration storage or storage in chilling temperature. So that is between 0 to 50 degrees Celsius, which we usually use for storing fruits, vegetables, meat, poultry, fresh milk and milk products, etc. And the last one is freezing temperature, so storing in the freezer. So that is between minus 18 degrees Celsius to 40 degrees Celsius, which we use for foods like frozen peas or mushrooms. So this is about low temperature method of hindering microorganisms growth. Next one is by drying of food commodity. So removal of water from the food to a level at which microorganisms fail to grow is an important method of preservation. So you all know that just like humans, microorganisms also require water for survival. So in this method we are removing that water to preserve the food. Moisture can be removed by the application of heat, as in sun drying and in mechanical drying. So this is the first type of drying. Okay, sun drying and mechanical drying. So another method of drying is by binding the moisture with addition of sugar or salt. So in this method what do we use? We use the process of osmosis, okay, where water moves from higher solute concentration, lower solute concentration to higher solute concentration. Okay, some examples are jams, jellies, or pickled mangoes, etc. Okay, the next method is by creating anaerobic condition. So anaerobic condition is nothing but condition wherein oxygen is not present. Okay, anaerobic condition can be created by removal or evacuation of air or oxygen from the package, replacement of air by carbon dioxide or inert gases like nitrogen. So anaerobic condition can be created by either completely removing the oxygen. So how do you completely removing the air? So how do you remove it? You create the vacuum. You completely remove the air and create the vacuum, or you just remove oxygen and replace it with other gases like carbon dioxide or nitrogen. Okay, so by using this method, what we can achieve is, first one, lack of oxygen prevents growth of any surviving bacteria and their spores under such conditions. So growth of any aerobic bacteria will be hindered by this method. Next one is production of carbon dioxide during fermentation makes the conditions anaerobic to prevent the growth of aerobes. So this is one such method where naturally the concentration of oxygen decreases and the concentration of carbon dioxide increases, which will stop the growth of aerobic bacteria. So another method is carbonation of drinks and storing fresh food under controlled atmosphere also serves the same purpose. So in carbonated drinks can be stored for long time without refrigeration because of that reason only, because it is carbonated and it does not contain oxygen in the package. Okay, next, in canned food also we remove the air by exhausting method. So this also will avoid the growth of aerobic organisms. And the last one is a layer of oil on top of any food prevents growth of microorganisms like molds and yeast by preventing exposure to air. So if there is a layer of oil in the food, in the packed food or in like pickles and all, so if you put a layer of oil, then the food material will not come in contact with the air which might contain oxygen. So this will not allow the growth of some microbes like molds and yeast. Okay, so next method of hindering the spoilage is by use of chemicals. Appropriate quantity of certain chemicals added to the food can hinder the undesirable spoilage in the food by two ways. Okay, one is by interfering with the cell membranes of the microorganisms, their enzyme activity or their genetic mechanism. So whatever chemicals we use, so that will interfere the metabolic processes or many activities of microorganisms. Next is it can act as antioxidant, so avoiding oxidation of any fat or any nutrients in the food. Okay, chemical preservatives are benzoic acid and its sodium salt, or sorbic acid, potassium metabisulfite and calcium propionate. Common antioxidants to check off flavor in edible oils include butyl hydroxyanisole, that is BHA, butyl hydroxytoluene, BHT, then tertiary butyl hydroxyquinone, TBHQ and then lecithin. So these are the antioxidants used. Okay, last one is addition of organic acids like citric, acetic and lactic acid in the food can inhibit the growth of many microorganisms. Okay, so then the next one is by killing the microorganisms. Okay, so killing can be done in two ways. One is by the use of heat and the other is by the use of radiation. So heat can be applied in different levels. So heating below 100 degrees Celsius is called as pasteurization, and next is boiling at around 100 degrees Celsius or heating above 100 degrees Celsius can be used for killing microorganisms. Let us see about killing the microorganisms using heat. So if we apply the heat, so it might cause coagulation of proteins, inactivation of metabolic enzymes, and finally destruction of microorganisms. Okay, so use of heat might cause coagulation of proteins and inactivation of enzymes in the microorganisms, finally causing the destruction of microorganisms. Exposure of food to high temperature also inactivates enzymes present in the food. Okay, like the browning causing enzymes like phenolases, so they are not they can cause food spoilage, right? So that's why heating can also inactivate such enzymes. Okay, next, food can be heated either at temperature below 100 degrees Celsius or around 100 degrees Celsius and above 100 degrees Celsius. Let us see more about these different methods of heating food. First one is pasteurization, where we heat the food below 100 degrees Celsius. So pasteurization is a mild heat treatment given to the food to kill most pathogenic microorganisms and is used in the food where drastic heat treatment cause undesirable changes in the food. Okay, for example, if you take milk, if you heat milk above 100 degrees Celsius, so it will cause the browning of the milk. Okay, so that is not the desirable in milk. So the caramelization of sugars in the milk can occur above around 80 degrees Celsius. That's why it is not desirable. So pasteurization is a suitable method for treating milk. Pasteurization is usually supplemented by other method to prolong shelf life. Pasteurization is most commonly used in treatment of milk and other dairy products, either as low temperature long time or high temperature short time process. So there are different methods of pasteurizing. So the first one is we use low temperature, but we pasteurize for long time. Okay, another method is high temperature is used for a short time. Okay, let us see the pasteurization temperature and time for different food. First one is heat treatment of milk can be done at 62.2 degrees Celsius for 30 minutes. So this is the low temperature long time process. Okay, so same milk we can do we can pasteurize using HTST, high temperature short time process, by heating the milk at 72 degrees Celsius and for 15 seconds. Okay, this is the most used method of pasteurizing milk. Then in pasteurization of grapevine, we use around 80 to 85 degrees Celsius of temperature for 1 minute. Okay, and beer is pasteurized at 60 degree Celsius. Pasteurization of juices depends upon their acidity and method of packing, whether in bulk or in bottle or in can. Bottled grape juice is pasteurized at 76.7 degrees Celsius for 30 minutes, while in bulk the juice is heated for 80 to 85 degrees Celsius for few seconds, okay, by flash treatment method. Then carbonated juices are heated at 65 degrees Celsius for 30 minutes in bottles, and vinegar in bulk is heated at 60 to 65 degrees Celsius for 30 minutes. Okay. Yeah. Let us see about boiling. Okay, boiling is nothing but heating at the boiling temperature of water, that is around 100 degrees Celsius. Cooking of food by boiling with water involves a temperature around 100 degrees Celsius. Boiling of food at 100 degrees Celsius kills all the vegetative cells and spores of yeast and molds and vegetative cells of bacteria. Many foods can be preserved by boiling. Example, milk, okay, which we commonly use at home, right? Even in canning, so canning of acid fruits and vegetables, we use boiling to kill the microorganisms. So some vegetables where we use boiling in canning is tomatoes, pineapples, peaches, cherries, etc. So here it is, the boiling is carried out at 100 degrees Celsius. Yeah, the last one is heating above 100 degrees Celsius. Heating by steam under pressure is used to obtain temperature above 100 degrees Celsius by using steam sterilizer or retort. So in retort, what do we use? We use steam under pressure. Okay. The temperature in the retort increases with increase in steam pressure. For canning of mushrooms and other non-acid vegetables, the processing temperature of around 121.1 degrees Celsius at 15 psi pressure are used. Okay, so this is the retort instruments used in industries. Okay, here they put the retorting foods inside the cans. Okay, then using the high pressure steam, they are retorted or sterilized. Yeah. So another method of high temperature heating is UHT, ultra high temperature process. So in this, the food is heated to a very high temperature at around 150 degrees Celsius. Okay, for only few seconds by the use of steam injection or steam infusion, followed by flash evaporation of the food of the condensed steam and rapid cooling. The process is also used for bulk processing of food. Yeah. For sterilization of milk and other liquid foods like juices, ultra high temperature process is used. So what do they do in ultra high temperature process? So they basically inject or infuse steam into the food. Like if you take milk, they inject the steam into the high temperature steam into the food, and then they flash evaporate whatever steam they have used. Okay, so they will remove all the steam they have infused. Okay, so this will ensure that the microorganisms are killed inside the food. Next is use of radiation for killing microorganisms. Irradiation consists of exposing the food to either electromagnetic or ionizing radiations to destroy the microorganisms present in the food. Okay, so this symbol you can see in radiated foods. Okay, irradiated foods will have this symbol. Examples of irradiations include use of ultraviolet lamps in sterilizing, slicing knives in bakeries. So here, this picture is little small. You can see in this chamber they use the ultraviolet lamps. Using the ultraviolet rays, they sterilize the knives in bakeries. Some other examples are gamma radiation from cobalt 60 or cesium 137 sources has been used for irradiation of many fruits like papaya, mango and onions, spices, fishes, etc. So for irradiation, basically we use solid foods. Okay, so liquid food irradiation is very rare. They are also used for inhibition of sprouting in onions and potatoes. So irradiations can also be used for inhibition of sprouting in onions and potatoes. Okay, so next method is prevention or delay of self decomposition of food. So it involves two stages, two types. First one is destruction or inactivation of food enzymes, and the second one is prevention or delay of purely chemical reactions in the food. Okay, let us see more about this. First one is by destruction or inactivation of food enzymes. Okay, so this involves two methods, that is blanching or boiling. Blanching is a mild heat treatment given to vegetables before canning, freezing or drying to prevent the self decomposition of food by destroying enzymes. Okay, so in blanching, what do they do? So once the fruit or the food is cut, so they dip or before cutting also, like leafy vegetables and all, so they dip into high temperature water or the boiled water for few minutes, for a minute, and they take it out. Okay, this will inactivate the enzymes in the food, which might cause decoloration or discoloration in the food. Blanching is carried out by dipping the food either in boiling water or by exposing it to the steam for few minutes, followed by immediate cooling. Okay, so they make it is made sure that the food is not cooked. Okay, just it is dipped and taken out and immediately cooled. So this is about the inactivation of food enzymes. Next is about prevention or delay of purely chemical reactions by using antioxidants to prevent oxidations. Foods containing oils and fat turn rancid and become unfit for consumption due to oxidation. Addition of appropriate quantity of antioxidants like butyl hydroxyanisole BHA, butyl hydroxytoluene BHT, tertiary butyl hydroxyquinone, that is TBHQ, lecithin, etc. are used, which can prevent the oxidation and can preserve the food. Okay, so remember all these antioxidant compounds. So we have studied chemicals, we have studied about these chemicals in the previous slides also. So these chemicals are used to prevent the oxidation in the food, which might cause rancidity in food. Okay, the last one is the prevention of damage by insects, animals, rodents and other mechanical causes. So for insects and animals or rodents, we can use the fumigants in foods like dried fruits, cereals, etc., which checks the damage caused by insects and rodents. Wrapping of fruits, providing cushioning trays, using light pack and good packaging material checks the damage to fresh food commodities during handling and transportation. So to avoid any mechanical stress to the food, we have to ensure the packaging and proper transportation of food. Okay, so this was all about the principles of preservation. Now let us see the methods of food preservation on the basis of food preservation principles. So in today's class we'll briefly look into the different methods of food preservation. Okay, so the different methods of food preservations include the physical method, then chemical method, fermentation, then combination method. So in physical method, we can remove the heat from the food or add heat or remove the water to preserve the food. In chemical method, so acids, salt, sugar or chemical preservatives are added to the food to preserve the food. Okay, let us see more about this. The examples of each type of preservation methods. Yeah. First one is physical method. So in physical method, by removal of heat, so we can preserve the food by using low temperature methods. So low temperature is basically you remove the heat from the food in order to get that low temperature. Okay, in refrigeration what do? You keep the food in the refrigerator. So the food is cooling down means you are removing the heat away from the food. Okay, even in freezing preservation, dehydrofreezing, then in carbonation, everywhere what do? We basically remove the heat making the food reach lower temperature and the food is preserved. Next one is by addition of heat. So in this method we use high temperature like pasteurization. So in that also low temperature long time pasteurization or high temperature short time pasteurization or sterilization, ultra high temperature processing and then microwaving. So these methods we have studied before also. So this is by heating the food. So by heating the food we kill the microorganisms. Okay. So next is by removing the water. So in this method we basically use drying. So drying can be of open sun, solar or polytunnel solar drying, and various other mechanical drying methods are also there. Then dehydration. So in dehydration, so we use mechanical drying methods. Then in evaporation or concentration, where we partially remove the water. Then freeze concentration, then reverse osmosis, then freeze drying, foam mat drying, puff drying, and various methods of drying are there to preserve the food by removing water. So in freeze drying what we do? So we freeze the food and then by creating vacuum we remove the frozen water molecules directly by subliming the waters. Okay, directly converting it to the vapors. Okay, yeah. Next is by irradiation. So in irradiation we use UV rays and gamma radiations to kill the microorganisms. So previously also we saw that cobalt 60 and cesium 137 is used for gamma irradiation. Then last type of physical method of food preservation is by non-thermal methods, that is using high pressure processing or pulsed electric fields. So in high pressure processing, so the food is kept in a chamber, okay, and then so high hydraulic pressure is given to the food, okay. So due to the pressure, the microorganisms inside the food will break out and they will be killed. So this is the HPP or high pressure processing method. And in pulsed electric fields, so what is done is, so pulses of electrons are sent, so electric field is given, so which will kill the microorganisms. So this is basically used for liquid foods. Okay. Yeah. Next is chemical methods of preservation. So in chemical methods we have different types. So the first one is by addition of acid. So basically the acetic acid or lactic acid is used. Example is pickling. In pickling of vegetables or like olives, cucumber, fish, meat, mangoes. So we use different types of acids to preserve the food. Okay, next is by addition of salt or brine. So in salted mangoes or vegetable slices and salted and cured fishes and meat, we use this salt or the brine solution in order to remove the water with the help of osmosis process. Okay, so in this also we have two types, dry salting and brining. Okay, dry salting is you just put some amount of salt on the food materials you want to dry. Okay, then you want to remove the water. So then in brining you dip the food into the brine solution, that is salt solution. Okay, the third method is by adding sugar along with heating. Okay, so sugar causes osmosis and heating kills the microorganisms. So this is used in confectionery products like jams, jellies, preserves, candies, marmalades, etc. Next one is by addition of chemical preservatives. So in this we have two types of preservatives. So use of class 2 preservatives like potassium metabisulfite, sodium benzoate, sorbic acid in food products is the first method of preservation by chemical preservatives. Then the second method is use of permitted and harmless substances of microbial origins like tyrosine, resin, niacin, as in which is used in dairy products for preservation. Okay, so the third type of preservation method is by fermentation. So fermentation can be alcoholic fermentation, which we commonly see in wine and beer. Okay, then the other type of fermentation is acetic acid fermentation, which is seen in vinegar. And then the other type is lactic acid fermentation, which is seen in curd, cheese, pickling of vegetables, etc. Okay, fermentation also increases the nutritive value of food. The last one is preservation of food by combination of methods. Okay, combination of one or more methods for synergistic preservation. So gives a better preservation and longer shelf life of the food. Pasteurization combined with low temperature preservation is one of the best examples we can see even in milk preservation. So what do we do with milk? So we pasteurize the milk first, and then we store it at low temperature. Then in canning, so heating is combined with packing in sealed container. So in canning, what is done? First it is boiled, as we studied before. So boiled, okay, boiled at around 100 degrees Celsius. The food is boiled, and then it is packed in a sealed container. So while packing also, the complete gases inside the can is exhausted, okay, to make sure that no oxygen is remaining inside the can. Okay, this is another example for combination method. So the last one is hurdle technology. Hurdle technology, like low pH, salting, addition of acid, use of sugar, humectant, and heating. So in hurdle technology, what we do is we create different types of hurdles for the microorganisms to grow in the food. Okay, so maybe it might be the combination of low pH. Okay, the organisms which can survive at low pH, so they will have to tackle the salt. Okay, the osmosis, salting will create the osmosis, right? So the low water activity is created. So the organisms which can survive here also, they'll have to face the addition of acid and the addition of sugar or humectant. Like this, various methods of preservations are combined in one single technology called hurdle technology. Okay, so this is all about the methods of food preservation based on the principles of food preservation. That's all for today. Bye bye. Continue your studies with iiocs.com Food Tech Club. So if you have any queries, you can contact the number on the screen. Thank you.